Okay, hi. Today we'll take our second lecture on DNA synthesis. We will describe and explain the DNA replication and as well describe the differences between the eukaryotic and prokaryotic DNA. The same reference is used, Molecular Cell Biology of Lodish, et al. Okay, so DNA needs to be copied precisely during DNA replication in order to prepare for cell division. This base pairing template model theoretically could be processed by either conservative mechanism or semi-conservative mechanism. Of course, there is a third mechanism which is called dispersive mechanism, but it's not so popular. For the conservative mechanism, the two daughter strands of the DNA forms new double-stranded DNA, and the parent duplex would remain intact. While for this semi-conservative mechanism, the parent strands are separated and each would form a duplex molecule with the newly synthesized daughter strand base paired to it. So actually, the experiment of Meselson-Stahl provided the evidence that the mechanism is by semi-conservative. So let's see what Meselson-Stahl's experiment states. So they initially grown bacteria in a medium containing a heavy nitrogen atom, which we donated, which is nitrogen 15. And as you know, nitrogen is a main component, this molecule is a main component in the formation of the nucleotide of the DNA. So the parent strand of the initial bacteria grown in that medium was all labeled with nitrogen. And this nitrogen actually, it's a kind of radioactive isotope of the nitrogen. So could you mute, please? Could you mute? So these bacteria grown in this medium containing the ammonium salt radioactive nitrogen 15 gave rise to DNA that is heavy labeled radioactive isotope. In case if the theory conservative is correct, so theoretically the second round of duplication, they grow the bacteria in another medium containing the normal nitrogen. So as we said, if theoretically it is correct, the conservative, you will soon have two bacteria from the parent one that have, one of them will have a bacteria with double-stranded heavy chains, and the other one, the new one, will be formed from the light chains. Since the new one have got its nitrogen or the ammonium salts for forming the nucleotide from the normal nitrogen. In a subsequent round, obviously, if it's grown in a normal media of nitrogen as well, nitrogen 14, you will see the old one exist as well, heavy, but you will see two populations of the light double strand. One population of bacteria will have this light, which came as a conservative from the parent, and the new light as well. Actually, basically, it's the same nitrogen 14, but they are formed new conservatively from the parents. So these, in case if a conservative mechanism apply. What about if it's a semi-conservative? Initially as well, it's grown in a radioactive nitrogen N15. It will get DNA double heavy strands. In the second doubling in a normal media, a new DNA is formed in the bacteria, which is composed of a strand from the parent and the new strand that is formed from the normal nitrogen. And hence we have a new duplex now, which will term it HL duplex, heavy light duplex.
Doctor?
Yes?
بس سوري أنا انقطعت عندي الكهرباء على 10 وهلأ لا قدرت أعمل جوين، فممكن إذا في شيء مهم يعني بس تعطينا هيك دغري summary.
You can go back to for the recording please.
بس هلأ كرمال كمل معك إنه أعرف عن شو عم تحكوا.
I'll say it again, okay.
Okay, thank you.
So you will have a heavy light, two daughter bacteria when grown in a normal media, that both of them will have a heavy strand and a light strand of DNA. In the next doubling, in a normal media of nitrogen, you will see again if it's theoretically according to the semi-conservative mechanism, you will see that the heavy light, each of them will give two daughter DNA molecule, and each of them will receive a template from the parent. So now, by the end, you will see a combination between double-stranded light and the double-stranded heavy light. So actually, these are the two mechanisms theoretically of the replication of a DNA done by Meselson-Stahl. Both of them are totally different, and both of them have distinct DNA. So if the case it's a conservative mechanism, so in the population of the bacteria, you will see some of the bacteria will have double-stranded that is having heavy nitrogen, HH. But if the case of the semi-conservative is the correct, you will see the heavy light double-stranded DNA in the population of dividing bacteria. So obviously, experimentally, we can differentiate or we can differentiate and we can prove which one is correct. And that's what Meselson-Stahl did. So to elaborate more on this experiment, so theoretically, in the first replication round of bacteria, if it's a semi-conservative, you can see here, you have a double strand that one of the strand is heavy N15 and one of the strand is light N14. And when you do a centrifugation, they'll migrate as one band. But in case, if it's a conservative, soon you will see two populations of a DNA in the bacteria. One of them is totally N14, and one of them is totally N15. And because N15 is higher in size, so it will migrate deeper in the centrifugation, so it's obvious two bands. So that's a predicted if the mechanism of replication is done either conservative or by semi-conservative.
Doctor?
Yes?
What is N15 and N14?
N15 and N15, every atom have a radioactive isotopes, okay? So the N14 is the normal nitrogen which exists in the periodic table. But some of these atoms, some of these molecules, sometimes have another state of excitation like N15. Okay, in this case, it can emit some radioactive properties and can be detected. Okay. That's why it's heavier when you do centrifugation, because it has N15 more electrons. Okay.
Yes, okay. Thank you.
You're welcome. For the second replication, as we see, let's go for the first round. You can see there are two populations, totally double-stranded normal DNA, totally double-stranded heavy DNA, HH, LL. Here you have a hybrid double-stranded DNA of N15 and N14, and this is a first replication. If you go back to the slide, actually that is correct, because in the conservative mechanism, theoretically, you have only HH and LL, while in the semi-conservative, you have only HL. That's why all of these are the same DNA, but here you have two different populations of DNA in the conservative. That's why it appeared here for the semi-conservative as a one band when centrifugation, while for the conservative appeared as a two bands. For the second replication, you can see Yes.
شو يعني لما نقول population من ال DNA؟
Sorry again.
شو يعني لما نقول population لل DNA؟ كنا عم نقول إنه في واحدة من two population واحدة بس one population لل DNA.
Yeah, bacteria that have this kind of DNA.
أوكي.
Okay.
دكتور
So-
شو الفرق بين الكونسرفتيف والسيمي كونسرفتيف؟ امتى بتصير كل وحده؟
We just describing theoretically. We will now prove which one of them is actually the correct one.
اوكي.
Okay. So for the second round, theoretically, if it goes by the conservative, you will still see both forms of DNA in the daughter bacteria. While if it goes with semi-conservative, you will see the same hybrid DNA in some of the population of bacteria, and as well you can see the normal double-stranded DNA. So theoretically, this is the cases if it goes with conservative and semi-conservative. But what about the actual observation that we got? When they experimented the replication, they saw that only the form of N15, N14 double hybrid, the hybrid double-stranded is only visible under actual observations. While after the second round, you can see as well the hybrid, and as well you can see the light double-stranded DNA. That's meaning that the correct theory of replication is the semi-conservative method. Okay?
Doctor,
Yes?
why in the second replication we got only N14? Yani one double strand N14 only. How did we get this?
If you go back to this figure, for the conservative mechanism, you're talking about which one, conservative or semi-conservative?
Semi-conservative.
Here,
About the semi-conservative mechanism.
the prediction or the actual?
The actual.
Okay, actually the actual for semi-conservative, it is the same as the prediction model. You can see it's N15, N14, it is the same. So why N15 and N14? Because as we said in this synopsis, you can see in the semi-conservative, upon the first round of replication, you can see that each strand, template strand from parent DNA, build up another new strand on top of this template strand. So we have a hybrid of H and L. That's why here we say N15 and N14. N15 for the H and N14 for the light L. Okay? So basically, you don't have any other population except H and L. Correct?
يا دكتور
Yes.
But
بس شو هي الجرين ستراند بالضبط؟
شو هي ايه؟
الجرين ستراند اخر شيء هو في
This
بلو
one-
ريد وجرين.
Okay,
Green.
the green strand means it is still light, but newly formed. Okay? It's different from the other parent double strand. Okay?
طب دكتور
But it's still
ما
light.
فوقها في وحده نيولي فورمد ما في فوقها وحده بلو وهي نيولي فورمد بس بلو.
Okay, let's clarify this one. Okay, that's me. It's a conservative mechanism, which is meaning that you have a DNA during replication. You will have a DNA that resembles the parent, and you can see this one resembles the parent, identified by the color. Both of them are blue, and you have a new double-stranded DNA that is formed. That's why it's a conservative. That's why there is blue that conserve the DNA from the parent, and there is a new one which basically formed irrelevant from the parent. So that's why it took another color for simplicity as green. But still, all these three are formed of the light nitrogen. Okay?
اوكي ميرسي دكتور.
You're welcome. So what actually the experiment that proved this theory, that the semi-conservative? Okay, let's recap on this slide. That basically just keep in mind, okay, that to identify a semi-conservative mechanism, the population after many times of doubling will only have two kinds of population of a DNA that is in the bacteria, two kinds of population. One of them is heavy light, and the other population will be light. So actually, the characteristic pattern of the DNA that characterize the semi-conservative is the presence of a double strand composed of a heavy strand and light strand, while the characteristic DNA of the conservative method is the presence of heavy double strand. So if the theory, to check the theory, if the semi-conservative is correct or the conservative is correct, you need to check which one of those is present in the doubling bacteria. So if you found that H and light is present, that means it's a semi-conservative mechanism. And if you find H at H is present, this means it is a conservative mechanism. So in the experiment, what they have did, okay. So actually this is density gradient. You can see to understand clearly, the band here, the black band here, represent the DNA migrating on the column or on the gel. You can see it's heavier toward this side, to the right side. While if it's on the blue line, that means it's lighter. So for instance, if the band migrate here, that means it's heavy. What if it's migrate on the blue line? That means it's light. What if it's migrate in the middle? That means it's an average between heavy and light, so it will migrate on the black line again. So you see the bacteria, they grow the bacteria actually at the zero. Just the bacteria have been grown in the medium, which, as you remember, provided by, as you remember here, the bacteria was initially grown in a medium containing N15. So all the media is heavy nitrogen. So here, yes.
Please, can you use a pointer?
انتوا شايفين الارو اللي انا بحركه ولا لا؟
لا.
Okay, hold on. Okay, is it obvious now?
Yes.
Okay.
Yes.
Yes.
Okay, so the bacteria was initially grown in the media containing heavy nitrogen. So that's why when it migrated over the gel, it shows a band here which corresponded to heavy. And as well, when they measured the density by spectrophotometer, they gave a peak at here at the region of heavy. What if after the first round, one means the first generation of growth? That means the bacteria have duplicated one time. After duplication one time, they realized the existence of the peak have been disappeared in the region of heavy, and the peak appeared in the region of heavy light. That means it is the beginning of the appearance of the hybrid double-stranded DNA that is composed of light nitrogen and heavy nitrogen. It is confirmed as well by the gel, because the band which was initially migrate at the region of the heavy, it now migrate at the region of the heavy light. And that's after the first duplication. After the second duplication, or the second generation of bacteria, you can notice that you have like two bands here. One of them is light, and one of them is heavy light. And the heavy band totally disappear. Confirmed by measuring the density, you can see that the peak have been split into two again. So that's mean after the first duplication, after the first duplication, only you can see heavy light DNA. But after the second duplication, only you can see heavy light DNA and light DNA. If you go back here, looking to the semi-conservative, you can see that after the first duplication, you only see heavy light. After the second duplication, you can only see a heavy light and the light. That's why it appeared here as a two peaks. One represented the light DNA and the other represented the heavy light DNA. What if after the third duplication time? After the third duplication of the bacteria, you will soon find that all of the fraction now is composed mainly of light and light, because the heavy have been consumed and there is no new source of new heavy nitrogen. So that's why after the third generation, a larger fraction of the DNA consists of light duplex. You have a very few HL duplex that soon will disappear, and the HH duplexes never appeared, because as I told you, in the semi-conservative, as we go back, as the semi-conservative, in the semi-conservative, you never see upon the division of the bacteria the presence of the heavy light, the heavy. So that proves that the main mechanism for replication in bacteria is, or generally, it is a semi-conservative. Going deeper of how the replication occurs. So DNA synthesis always proceed in the 5-3- direction. The polymerases, the DNA polymerases, cannot initiate synthesis de novo. What's meaning de novo? They cannot sit on the DNA by themselves and do the replication. They need a guidance for enabling these DNA polymerases to sit on the DNA to do polymerization and replication. So for this case, they require a short RNA or DNA strand, which is called the primer, to begin the chain growth and replication. So actually, they need this guide, the primer, to tell the polymerase where to start the replication. Helicases are also involved for unwinding and melting of the parent DNA in order for the duplex DNA to function as a template during replication. The unwinding begins at segments in the DNA molecule, the replication origins, and usually these replication origins are AT-rich region. At these replication origins, a replication fork is formed. Once helicases unwound the parent DNA molecule, RNA polymerase, which is called the primase, forms a short to around 12 nucleotide sequence. Nucleotides, it's called RNA primer, which is complementary to template strand. And as we said before, this primer is crucial for identifying the location that the DNA polymerase will start initiating the process of replication. Since the DNA polymerase cannot initiate this process of synthesis de novo by itself, the primer is elongated by using a specific DNA polymerase alpha, for another 25 nucleotides, forming a primer made of RNA at its 5' end and the DNA at its 3' end. So basically, the primer which is formed to guide the DNA polymerase to build new strand is formed from a part that is made by RNA polymerase called the primase, and the part is made by DNA polymerase alpha. Okay. So the local unwinding of duplex DNA produce, if you remember from last lecture, the torsional stress, which is relieved by the topoisomerase 1. Let's see what's actually happening. So you have now a double-stranded DNA, and of course it is anti-parallel. You have the helicase, which starts unwinding of the DNA. Once it's open, the replication fork appears here, and the helicase is going on doing its job. After that, you have two template strands. This template strand, the primer is initially made on this template strand, and the polymerase start to work in this direction. As we said, always DNA replication occur in the direction 5', 3'. So again, the helicase start to work, the primer is there, the DNA polymerase start to do its job from the 5' away to 3'. And this, you notice that this strand is replicated continuously. That's why this strand is called the leading strand. Again, so you can see this strand is continuous in replication, and this strand is called the leading strand. Here there is a problem in the second strand, because actually the second strand is reversed, and replication had to be done in the direction of 5', 3'. So how the replication try to solve this, the replication machinery try to solve this issue? In this case, when the fork is open, the primase start to put its primer here, and the DNA polymerase will start doing its job and doing the polymerization. And it needs to move from direction 5' to 3' in that direction. But this is a problem, because when the fork is going deeper, another template strand is open. So a new primer is laid on the DNA, and the polymerase is doing its job of polymerization, and this process is repeated. The result of this, you will have fragments of DNA. Each fragment will have a primer at the front of it at the 5' end, and the part of the daughter strand DNA. And this fragment is discovered and named according to a Japanese scientist, so it's called Okazaki fragment. So basically, the Okazaki fragments, actually it's regions of the DNA that consist of the primer and a part of the newly synthesized or formed DNA. Soon after that, enzyme called a DNA ligase ligate each of these Okazaki fragments to form a complete strand. And as well, enzymes are used to remove these primers, because it's not correct to leave this primer sequence in the sequence of the newly formed strand. So since this strand is formed discontinuously, that's why this strand is called the lagging strand. So we have now two daughter strands have been formed on the template strands of the paper, of the parent. One of them have been formed continuously, no obstacles, since it is continued from the 5' until 3'. The other one have been formed discontinuously, then rejoined by DNA ligase. That's why the strand which eventually will form will be named lagging strand. Keep in mind that polymerase zeta, polymerase epsilon form the leading strand, while polymerase delta replaces the ribonucleotide. Of course, where is the ribonucleotide of the primers? With deoxy ribonucleotides to form the lagging strand.
دكتور
Yes.
فيك تحكي لنا بليز عن الفورك باس؟
عن
الفورك موفمنت.
الفورك movement, it is as a result of the action of the helicase. It melts DNA, so it opens up the DNA, okay? So this place actually, as you can see from here, this is the replication fork, where it located, okay?
يس اوكي مش الحال. شكرا كثير.
طب
دكتور؟
الهليكيز
Yes.
الهليكيز كيف بتعرف لوين بس بدها تفتح ال DNA strand؟
Sorry again.
الهيليكيز مش هي اللي بتفتح ال DNA؟
Yes. Yes.
كيف
The
بتعرف
helicases open melts the hydrogen bond for opening the DNA.
كيف بتعرف لوين بدها تفتحها؟
There is a lot of other mechanisms that guide the helicase through the replication origin. Okay.
اوكي طيب دكتور بس سؤال كمان ليش الستراند اللي تحت لما عملت duplication كانت leading strand اما اللي فوق ما كانت continuous؟
ما كانت continuous because as you know here the replication occurs from 5 prime straight to 3 prime since the template already is reversed. Okay. ده في مشكلة؟
لا تمام.
Okay, but once you open this strand is different because the terminal end is 5 prime and the polymerization had to start from the 5 prime, so it cannot start from here as in this case. Okay, because this end of the new formed strand will be 3 prime and it cannot move this way. Okay, it cannot move from 3 prime to 5 prime, so it had to move from 5 prime to 3 prime. So in this case, as you can see, when the fork starts to open, after the helicase melting of the hydrogen bonds of the DNA, you can see it starts to open a little. So a new primer is added here, and starts to build on polymerization. And when it starts again, when it's open deeper by the helicase, another primer is added here and it moves in the other direction. And when the helicase goes deeper, another primer is added here and it goes in the opposite direction. The result of this, you have three discontinuous Okazaki fragments because of the way and the orientation of the replication from 5 prime to 3 prime. Okay?
طب دكتور اللي فوق على الستراند الفوقانية بتكون هي RNA nucleotide مش DNA؟
These red ones are RNA nucleotide due to the action of RNA polymerase. That's why the polymerase delta, after the ligase, ligating all these Okazaki fragments, the DNA polymerase delta had to replace this ribonucleotide, which is represented by the red, with deoxynucleotide. Okay?
Okay.
بس كأنه تحت بقى في عنا RNA.
Sorry?
كأنه تحت بقى في عنا short RNA primer ولا لا؟ على الستراند اللي تحت.
Here?
Yeah. Yes.
Yeah, there is a short RNA primer, but it is only one at the beginning. Okay?
Okay.
And it will be removed as well.
Okay.
So هن in total 4 primers?
Sorry?
هن in total 4 primers are added to the DNA?
No, you can't say 4 or 3 or something. It depends on the length of the DNA.
Okay, thank you.
دكتور امتى بيكون اسمها lagging strand؟
Lagging strand, you have only 2 names, either leading for this one or lagging strand for this one. When you see discontinuous formation of the daughter strand through the formation of Okazaki fragment, in this case it's called lagging strand. Okay?
Okay, sir.
Okay. So I'm reminding you, so we have a major problem if you remember that we have said here that due to this unwinding of duplex helix, a torsion stress arises, which is relieved by the topoisomerase. I'm reminding you that there are 2 enzymes. We have said that in the last lecture. We have two enzymes responsible of doing this relieving of the supercoils or torsional stress. And these are important actually to relieve the DNA during the process of coiling or during the process of replication. Okay? So here you can see another figure representing the DNA replication. And you can see here, this is the double strand. These are the action of the helix opening up the double strand by melting the hydrogen bonds, and this creates a replication fork. You can see while doing the opening or the melting of the DNA at the other end is coiled. So it needs the action of the topoisomerase, which stays here and relieves this torsion stress in order for the helicase to move smoothly on the DNA to melt the hydrogen bonds. Once the DNA have been melted into single strand, there is another important set of proteins which is called single strand binding proteins. These single strand binding proteins will help that these single strands DNA don't re-anneal and bind to each other again. And you can see here how the leading strand is formed from the 5' straight away, and again how is the lagging strand is formed. You can see here, this is the action of the ligase, which sits on the fragments and the try to ligate Okazaki fragments. And you can see as well how the primase is sitting for forming the primer that will guide the DNA polymerase to move. So basically, this figure is basically the same figure as the one we have just said in the last two slides. The only difference that it shows the presence of two important proteins, the topoisomerase for relieving the torsional stress and supercoiling at that end of the DNA. And you can see as well the single-stranded binding protein, which the main function of this protein to keep the single strand from binding or re-annealing again, or from forming the hydrogen bond. Once the replication is finished, this single-strand binding protein will leave the template DNA, enabling the DNA to rejoin. So actually, this is an animated figure to see the process is extremely complicated, as you can see. So actually, you can see how the process is occurring, and of course, it's very fast. Okay. Actually, this is a simulation of the complex process of replication by a semi-conservative mechanism. Okay. So basically, the replication, it's not occurring only in one direction. It occurs in bidirectional from one origin. So normally we have two replication forks. Okay. So normally we have two replication forks. They assemble at one single origin of replication and then move into opposite direction, leading to what is called bidirectional growth or replication of both daughter strands. So actually the eukaryotic chromosomes, DNA chromosome, have a multiple replication origin. So you can see here, for example, this is a circular DNA. When it's cut with an enzyme called a restriction enzyme, EcoR1, this enzyme converts the circular DNA. Let's use the pointer. This enzyme will convert the circular DNA into linear DNA, as you can see, linear DNA. And we assume that this is the replication origin that occur and the start of replication. So you can see that at that location, the DNA melts and the hydrogen bonds are melted. So a replication bubble arises that soon become larger for building up the daughter strand on the temple strand. So you can see this in the diagrammatic sketch. While here, actually, what's really happening under the electron microscope. So you can see the replication bubble, which is formed actually of the template strands of the parent, and it will be used to build the daughter strand. to describe more what is bidirectional mechanism, so you can see here it is a linear DNA, double-stranded DNA. The helicase sits and binds to the DNA at the replication origin. It melts DNA, opening up DNA by process of unwinding and melting the hydrogen bonds. Soon after that, the replication starts. So here you can see the beginning of the formation of the two daughter strands on each of the template strand of the parent, while the helicase is moving this way and this way in opposite ways, trying to open up more in the DNA. You can notice that the initial part of the daughter formed strand is red, which reflects the presence of the primer. Because as we said before, the DNA polymerase needs a primer to identify the location to start the replication. Soon after that, the helicase moves further to opposite sides, opening up DNA, enlarging the bubble of replication. And now the replication continues in this direction. And keep in mind that replication occurs at the direction of 5', 3'. Soon after that, the leading strand is formed on both sides, since it's a bidirectional. Again, when the helicase moves in another direction, in opposite direction, and opening up more DNA, a new primer is being laid here or formed, which enables the formation of the new polymerization of the strand. And soon after that, there is a ligation between these two strands to form the complete daughter strand. Actually, this is a simulation of the complex process of replication by a semi-conservative mechanism, okay? So basically, the replication, it's not occurring only in one direction. It occurs in bidirectional from one origin. So normally we have two replication forks, okay? So normally we have two replication forks. They assemble at one single origin of replication and then move into opposite direction, leading to what is called a bidirectional cross or replication of both daughter strands. So actually the eukaryotic chromosomes, DNA chromosome, have a multiple replication origin. So you can see here, for example, this is a circular DNA. When it's cut with an enzyme called a restriction enzyme, EcoR1, this enzyme converts the circular DNA. Let's use the pointer. This enzyme will convert the circular DNA into linear DNA, as you can see, linear DNA. And we assume that this is the replication origin that occur and start of replication. So you can see that at that location, the DNA melts and the hydrogen bonds are melted. So a replication bubble arises that soon become larger for building up the daughter strand on the temple strand. So you can see this in the diagrammatic sketch. While here, actually, what's really happening under the electron microscope. So you can see the replication bubble, which is formed actually of the template strands of the parent, and it will be used to build the daughter strand. To describe more what is bidirectional mechanism, so you can see here it is a linear DNA, double-stranded DNA. The helicase sits and binds to the DNA at the replication origin. It melts DNA, opening up DNA by process of unwinding and melting the hydrogen bonds. Soon after that, the replication starts. So here you can see the beginning of the formation of the two daughter strands on each of the template strand of the parent, while the helicase is moving this way and this way in opposite ways, trying to open up more in the DNA. You can notice that the initial part of the daughter formed strand is red, which reflects the presence of the primer. Because as we said before, the DNA polymerase needs a primer to identify the location to start the replication. Soon after that, the helicase moves further to opposite sides, opening up DNA, enlarging the bubble of replication. And now the replication continues in this direction. And keep in mind that replication occurs at the direction of 5', 3'. Soon after that, the leading strand is formed on both sides, since it's a bidirectional. Again, when the helicase moves in another direction, in opposite direction, and opening up more DNA, a new primer is being laid here or formed, which enables the formation of the new polymerization of the strand. And soon after that, there is a ligation between these two strands to form the complete daughter strand. So we will now talk about in vitro DNA replication. So what's in vitro DNA replication? That means how you can do replication of a DNA in the lab. So what we have talked in the last few slides, actually what is happening inside our cells, in vivo, inside our cells. But now we are talking how we can mimic the process of replication in a test tube, in a lab, in vitro. Okay. So basically, there's another name for that process. We call it PCR, or polymerase chain reaction. So to make a polymerase chain reaction, you basically need 4 components. You need the double-stranded DNA, that's meaning the parent DNA. You need excess of both primers, and of course, you know the importance of the primers, because the primers guide the polymerase to the start site of replication. And of course, you need the raw materials, which is the 4 nucleotides, A, T, C, G. And of course, the most important one is the polymerase, which here we normally use Taq. It's named Taq polymerase. And it must be heat stable, as we will see now. So this Taq polymerase, which basically it's a DNA polymerase, is used for the process of polymerization and the replication of DNA in vitro, or PCR. So initially, you have a double-stranded DNA, as you can see. Here, you don't have any helicase as normal, as we have in our body cells. But we use instead temperature at around 95 degrees Celsius to break down bonds, hydrogen bonds, and this lead to a breaking down of the hydrogen bond, and you have now single-stranded parent strand. Then you include the primers. The primers will anneal at a specific location. So normally we have a pair of primer which will anneal as in the figure, and you need a temperature of around 50 to 60 degrees Celsius for the primers to anneal. And now the primers are set at the location you need to amplify or replicate by the Taq polymerase. Soon after that, you lower the temperature to 72 degrees Celsius to enable the formation of the process of polymerization. And you can see that the Taq polymerase will start doing the job and will build the DNA daughter strand above the template on both parent strands. And now you have 2 molecules have been produced in the test tube from a parent molecule. What actually you need to do now is to repeat the process to have a huge number of molecules starting from just 1 molecule of DNA. So again, you denature the DNA, same temperature, you anneal the primer for the 2-1, leave time for elongation of the primer or the polymerization or the replication, and now you finished cycle 2. You repeat this for cycle 3, you are now having 4 molecules. For cycle 3, you have 8 molecules, and repeating that over a large number of cycles that can reach to 20 cycles. This will amplify DNA sequence to a 1 million fold. Okay? Prokaryotic DNA. Okay, in the last part of the lecture, we'll talk about the difference between prokaryotic DNA and eukaryotic DNA. So generally, the prokaryotic DNA is circular DNA, single circular DNA. You can see it's a double-stranded, but it is circular. But in case in eukaryotic DNA, it is a linear DNA. As you can see, it's a linear DNA. Okay, you can see that the circular DNA of the prokaryotes, like bacteria, can be attached to the cell membrane, which is not the case in the eukaryotic DNA. Cannot be attached to the cell membrane because it is confined inside a nucleus. And of course, you can see that the prokaryotic DNA exists inside a nucleus. But here, there is no nucleus, of course, for the prokaryotes. The second, you can see here, the second major difference that in prokaryotes, you can see they have 2 types of DNA. You have chromosomal DNA, and you have something which is called extra chromosomal DNA. We normally don't say extra chromosomal DNA, and we say it plasmid. And here you can see electron micrograph to show the structure, the shape of the real plasmid. And you can see it's how heavily supercoiled and can exist in multiple forms. And as well, plasmid can exist in many numbers. It's not only one molecule of plasmid DNA in the bacteria. Again, you can see here how the difference between the DNA of the bacteria and and an example of a eukaryotic DNA. That's not our DNA, okay? It's an example of eukaryotic DNA. So you can see how it's here it looks thin, but here it looks thick DNA. So majorly, what is the reason of the thickness of the DNA in our in eukaryotic DNA? Because normally in eukaryotic DNA, it is combined with histones and the proteins. So that's why it appears thicker under the electron microscope. Again, here you can see in prokaryote, because there is no nucleus, the DNA wants to do a process of a transcription. We will deal with transcription in the coming lecture. Once the DNA do transcription, which is the process of formation of a messenger RNA, this messenger RNA after transcription can undergo protein synthesis straight away, simultaneously, to form a protein, because there is no nucleus. But the process is complex in the eukaryotes, because the DNA, when after transcription, this messenger RNA exists inside the nucleus and have to migrate to the cytoplasm to undergo translation for the protein. So basically, the process of transcription, which is the formation of messenger RNA, and translation, which is the formation of proteins, transcription and the translation in prokaryotes are coupled, can occur at the same time due to the absence of the nucleus. But in the eukaryotes, they are separated because transcription occurs in the nucleus and the translation occurs in the cytoplasm after the migration of the messenger RNA from the nucleus to the cytoplasm. As well, you can see a major difference between prokaryotic DNA and eukaryotic DNA. You can see in the prokaryotic DNA, it have only one origin of replication, while for eukaryotic DNA, you can see a multiple origin of replication. So that's mean that the prokaryotic DNA start replication from only one side, while for the eukaryotic DNA, it have multiple sites for origination of replication. Again, if you can see, these are the plasmid, the circular DNA, and these are the linear eukaryotic DNA. You can see that in the circular DNA, there is only one origin of replication. But here, in the linear DNA, you can see multiple origins of replication. So in this case, The helicase open these origins of replication, so we'll have a lot of transcription bubbles. But here only one bubble because there is only one origin of replication. So again, you'll have here one replication bubble and the two replication forks. But here you can see multiple replication bubble and forks. Soon after that, the process of replication occur bidirectionally, as we already have said. But here you can see only one bidirectional replication, and it will cover all the circular DNA until the end. But here you can see the bidirectional replication occur until replicating the whole linear DNA. Finally, you get two circular DNA, and you get here two linear DNA. Here we'll have like a summary for all the comparison between the prokaryotic DNA and eukaryotic DNA. For the prokaryotic, it's a single DNA, multiple DNA for eukaryotic, have plasmid, no plasmid in eukaryotic. Of course, circular prokaryotic DNA, while linear, the DNA in eukaryotic is linear. The DNA here in the prokaryote is in the cytoplasm, normally in a region called nucleoid. It's not a nucleus, it's a region in the cytoplasm which is the DNA is confined at that region. And the condensation of the DNA of the prokaryotes is via supercoiling, while the case is different in the eukaryotes because it's confined, the DNA is confined into the nucleus, and the condensation, as we said in the first lecture, occurs by histones. Of course, the DNA here naked, there is no histones, but here DNA is associated with histones. The genes in the prokaryotes are haploid, but of course we all know that the genes in the eukaryotes are diploid. For prokaryotic DNA, there is little repetitive DNA. And as we said in the last lecture, DNA of eukaryotes have large amount of non-coding and repetitive DNA. Genes for prokaryotic DNA do not contain introns. Introns are regions of non-coding, but here in the eukaryotes you have a lot of introns, and we will deal with introns in the fourth lecture. Genome here is organized in operons, which is a functional unit for gene expression, but here usually don't exist in eukaryotic DNA, and as well we will deal with operons in the next lectures. Here, the non-essential genes are encoded by the plasmids, if you remember, the extra chromosomal DNA. But of course, in eukaryotes, there are no plasmids. As we said, in prokaryotic DNA, there is one replication origin, but in the eukaryotic DNA, there is multiple replication origin. Of course, as we said, because DNA interact directly with the cytoplasm and they exist in the cytoplasm, so transcription and translation occur simultaneously, coupled together in the same time can occur. But in eukaryotic DNA, the story is different because transcription occur in the nucleus and the translation occur in the cytoplasm. And here you can see the reference pages from the same reference for DNA synthesis. And as well, I give you the reference for the next lecture for RNA synthesis, transcription, and processing from chapter 5 and chapter 9. And this concludes our lecture. I'm waiting for your questions.